Okay, why don't we get started then? So thank you all for joining and coming to the TNT Network seminar, the first of our new series. Um, for those who are kind of joining for the first time, TNT is the Translational Neuroengineering Technologies Network, and we're a kind of interdisciplinary, interdepartment uh, student organization here at Hopkins for anyone interested. Obviously, as you can tell by the name, in translational neuroengineering technologies. And Yeah, we're excited to have you guys here and hear from Dr. Fridman, who is kind of kicking off this new seminar series. Dr. Fridman is a professor in the departments of otolaryngology and biomedical engineering, and he is also the faculty advisor for the TNT group. So we know him very well, and he's been a great mentor for us. And so he'll get us going with this talk today, talking about currently unsolved problems in neuroengineering. In terms of the talk today, Gene said he is planning on speaking for about 45 minutes to an hour, but you guys are welcome to interrupt during the talk if you guys have questions of clarification or interest or whatever. So feel free to do that. And so we'll get started. One other thing, after the talk, we'll have a poll for you guys to fill out. So we'd really appreciate if you guys stick around to the end. The poll basically is to help us get information about who's attending and what you guys thought about the seminar. So with that, I think we are ready to go. Gene, please take it away.
Cool. Good thing. Thanks, Mark. Okay, well, I'm excited to deliver this inaugural talk for this year. All right, so and I thought this would be an appropriate topic for sort of the initial initial session. So currently unsolved problems in neuroengineering is what I kind of chose. So I wanted to focus specifically on commercial devices because there's a lot of research going on in neuroengineering, but really when the technology becomes viable enough, it starts to get to translation. And so I wanted to kind of focus on that and leave the research to the end to kind of talk about how problems are getting mitigated. So first I want to start with the fact that evoking neural activity is very effective and it's very easy to do with electricity. And this is what I typically suggest that my students do in the lab, because really it's kind of indicative of how easy it is to evoke neural activity somewhere. And if you try this, you'll get a little shock on your tongue. You're not going to be doing this with your eye. But you'll see how easy it is, and you'll see that you actually get a sensation. You get a sensation of acid, and you know exactly where that stimulus is going, even though it's not pattern, it's not, there's nothing special going on. Okay, so there's a minor safety complication with this whole concept, because if I keep doing that, right, what will happen if I put a battery in a cup of water? So think about your body being a cup of water, and you take that battery and you stick it into water. If you wait for a while, what you're gonna see is this. You're gonna see electrochemical reactions. You're gonna see electrolysis. Now, this is of course not very good if these electrodes are implanted in the body. And so for this reason, then the implantable devices don't use this kind of electric current. Instead, what these devices do is they deliver what's called charge balanced biphasic pulses. So the idea is that you have an electrode, and then you deliver some electrons to one of the electrodes, and then you remove those electrons before they have a chance to jump over into the solution and cause a chemical reaction. So, so all of these neural implants use charge balanced biphasic pulses, and this device that you see here is called implantable pulse generator, or an IPG is the technical term. And so what you'll see is various applications of this IPG because it's been used in many, many devices. So the history of neurostimulation started, of course, a long time ago with Galvani and with Volta in the 1800s, but those experiments were just experiments, and of course, you know, those were just sort of prototypical kinds of experiments. But then the first actual stimulator, first actual commercial device was made in, I believe this was in Karolinska Institute, where they hand out Nobel Prizes. And this was a pacemaker, and this was a fairly simple device. It would deliver a pulse every so many milliseconds, and it had an internal timing mechanism that would just simply keep track of that and just would deliver a pulse every every however many hundreds of milliseconds. Okay, so that was the first one. And now we're going to kind of go through and look at other ones that have been done since and see what's been done. So the fundamental technology, like I said, is an implantable pulse generator. Sorry. Okay. So this kind of an implantable pulse generator, and they all look similar. It's basically a can that doesn't have any sharp edges on it, so that it kind of fits into the body and doesn't really cause any reactions, immune reactions, and is sort of nicely integrated into the body. And then it has a lead with wires in it, and at the end of this lead, you have exposed metal contacts, and those are called electrodes. And so one thing you can notice here is all of these devices, this one's for chronic pain, this one's for urinary incontinence, Parkinson's disease that goes into the brain, and then cochlear implants, they all look approximately the same. And as far as the patterns that they put out, again, these are biphasic charge balance pulses. And the general assumption when you first kind of start looking at these devices is they evoke, at least for some range of pulse rates, they would evoke one action potential per pulse. And so here you see this is from a cochlear nerve for a cochlear implant. You see this is a PSTH. It's a histogram of action potentials as you deliver these pulses. Okay. So 65 years later, we have a lot of neural implants out there. I am going to focus on 3 specific ones as a case study, because what I wanted to do for this talk is to look for common problems. And so I chose these 3. I chose the retinal implant because it's sort of mid-range as far as its history is concerned. It's been out for about 15 years. And so we can look at how well it's functioning and the problems associated with it. The other one I wanted to look at is a cochlear implant. This is probably the most mature technology that we have. And it's been out for 50 years. So the initial ones came out in late 60s. And finally, I wanted to look at the vestibular implant. This is, of course, a major point of work here at Hopkins. And this has been out for about 5 years. So I've been involved with this one also. So that's why I wanted to talk about these 3 to kind of give you a range of these technologies. Okay, so let's look at the retinal prosthesis first. So it's kind of a basic physiology. You have your eye, and then you have your retina. Retina is this tissue that's on the back of the eye, and there is some processing going on. You have these photoreceptors that are sensitive to light. Then there's some processing going on here with bipolar and horizontal cells. And then And then at the end of that processing, you have retinal ganglion cells. And these cells get some feedback, they get some information from all of these, from a number of these photoreceptors. And then they kind of integrate them together, and then they respond accordingly for each ganglion cell. And then the result then flows down the axon and actually goes down the side on the outside of the retina, and then down the optic nerve. Now, this is kind of important, the orientation of the retina. So the light actually, if you notice, comes in through the retina all the way through before it actually ends up exciting the photoreceptors, and the processing actually happens closer to away from the edge of the retina. Okay, that's going to become important in a minute. So here's a retinal prosthesis. This is, again, probably the most mature one. This is Argus 2, I believe, and this is from Second Sight. This was started probably 20 years ago. It didn't actually come out to the market, maybe 10 to 15 years ago. This is what it looks like, the end of this thing. Again, you have a can that is that's going to do the processing, and it's connected to a coil that's going to be able to receive information from the outside. And it's sitting on the retina. It's an epiretinal prosthesis, meaning it's sitting on the side of the ganglion cells. So it's going to be stimulating from the side of the ganglion cells. Now, there are others, but they're less mature. So there's subretinal prosthesis, and then another one is called suprachoroidal. I'm going to focus on this epiretinal again because it's the most mature technology. So here's what we get. How well does it work now? And I specifically for this talk, I wanted to pull up the latest reviews. And so, so one, this is sort of investigating what is it that these subjects see when you stimulate on an individual electrode. So these are different subjects on each row, and then D2 denotes the row and the column of the individual electrode. So they stimulate on that electrode, then they ask the subject, what do they see? And have them describe it or draw it, and so on. And so these are the kinds of, and these are different trials, which is also interesting, that they're not necessarily consistent, but this is what you see. You see these lines, you don't see dots, you see lines, and some of them are fat, some of them are thin. Now, and they're all oriented kind of in specific directions. So in the end, then, what is it that this device does? It's been out for about 15 years. How well does it work? Well, what we have is phosphine shapes have been reported to be round dots, dark centered rings, complex arcs, blobs, or ellipses. But they are in approximately the correct spatial locations, right? So you know that this is, you can sort of scan to see what you're looking at. Now, these phosphines are large. They're between 1 and 10 degrees. So you can kind of see 10 degrees as if you take your hand and you put it at the full extent, that's going to be about 10 degrees span of your retina. And so your thumb is about 1 degree or something like that. So these are big blobs that you see, and the brightness of these blobs is going to be modulated to about 10 gray levels. That wasn't me. Who was it? Okay. So, all right. So, I mean, this is not great for if we're trying to replace the vision for somebody. This is not fidelity. This is a basic start. But again, 15 years later, you would expect, or I don't know. Without knowing the topic, I guess I would expect more. All right, so let's find out why. Let's kind of focus on what's going on here. So retinal ganglion cells, what they do is they respond to edges. And so if we're stimulating these retinal ganglion cells, what we would like to do is we would like to deliver the sensation that or the information that is already processed by the retina itself. And that's what we see here. And our technology as it is now is really, really good at being able to figure this out. This is not difficult to figure out how you can edge detect this image. This is sort of a common thing that's done in image processing. This is not a difficult thing to do. And so this is the kind of information that we want to send to the brain. Okay, now here's a bit of complexity. So the retinal ganglion cells, these are there are multiple types. The most basic ones are on center and off center. Now, what does that mean? It means that if you shine a dark light on this particular area that the ganglion cell is looking at, then you're going to see some spiking activity. If you shine a little bright dot right in the center of that field, it's going to respond really, really well. Okay. If you shine a dark light, it's going to stop firing. Okay, at the bottom here. If you give it a full kind of view, then it's going to fire maybe a little bit more than with dark, but it's kind of in between. So off-center cell responds exactly the opposite way, right? You can see if you shine a bright light, it stops firing. If you shine a dark light, if you shine a light on the surround of this thing, then it fires more. Now, to add to the complexity, there are these cells are divided into different types also, and some of them fire in a sustained fashion for a while after you add on a stimulus, and some of them fire only at the onset of these. So this is a complicated thing. Now imagine that you have, you're looking at a couple of these cells. One, let's say, is an on-center off-surround. The other one is off-center on-surround. And you can see as these cells kind of go from a light area to dark area, you can kind of see their response. So as this particular on-center off-surround is encountering a dark stripe, it will fire more, right? But in the same sense, this other one that's sitting here is going to fire less in response to that being basically placed in that same location. And exactly the opposite happens here when the on-center gets into the dark place and the off-center gets into the same dark position. Okay. Now, this has been known for a long time. These patterns of innervation, or these patterns of these ganglion cells, are a mosaic throughout the entire retina. They literally tile the whole retina. So when you talk to an individual cell, when you record from an individual cell, you don't know whether it's an on cell or an off cell. You don't know whether it's transient or sustained in any one location. And they're all sort of tightly interwound in every location on the retina. Okay, so the added difficulty here is when you're stimulating something called axons of passage. So what you see again is in the retina, if you remember the axons from all of the ganglion cells, they all go into the middle part of the retina, and they go down the optic nerve. And so at every location on the actual retina, not only do you have the individual bodies of the ganglion cells, but you also have these axons that come near it from different locations. And so that is what basically accounts for these streaks that people see. And what's happening here is they're comparing what happens with let's say, a large electrode versus a small electrode. So the effect is basically the same, except for small electrodes, you have smaller streaks and they're smaller dots if you can get them. Okay, so here's the bottom line as I see it for the retinal prosthesis. On a microanatomic scale, the cells that need to be stimulated carry complex coding, right? It's not very simple. There's an on and an off reaction, and then that's a temporal reaction, and there is a reaction to whether or not it's dark or light. And then you have these on, sustained, transient, and as I was looking it up, it turns out there's like 30 different types that are known, that are different sizes, different sensitivities, and different temporal reactions. And so all of these are not spatially distinct, they're all tiled. So the bottom line is the prosthesis needs to distinguish between these cell types, and these cell types really, in order for us to get the fidelity that we need, they really need to send the proper information to the brain if we're going to be stimulating these things. So the prosthesis really needs to distinguish between these cell types and stimulate them selectively with appropriate stimulation patterns if we're going to get the fidelity that we want at the brain. Okay. Now I'm going to go on to cochlear implants, and let's look at that. So this is the cochlea. So when you're hearing, you have the outer ear, you have your middle ear that amplifies the sound. And what that does is when the signal comes in from the outer ear, this vibrates a little window in the cochlea, this snail-shaped organ, and this vibration causes a vibration along this membrane that bisects this entire organ, and this membrane is called a basilar membrane. And if you put in a particular tone or frequency of sound, it will vibrate a different location along this membrane. So high frequencies are at the basal end, and then the low frequencies are closer to what's called the apical end, the end of that snail shape. Okay, so the neurons are located along this membrane, and so the brain knows which frequency was heard based on which neuron was excited. Okay, so that seems phenomenal for neuromodulation, because if you stick that battery in your tongue, your tongue is getting stimulation and not your foot. And so same deal here. If you presumably stick electrical stimulation on closer to the basal end, presumably the brain will interpret that as low frequency. And if you stimulate closer to the apical end, presumably it will interpret it as being high frequency. The loudness is interpreted based on how many neurons are excited in a given location. Okay, so this was the basis for building an auditory prosthesis. Okay, so this is a drawing of a lead, and again, you have electrodes positioned along this lead. And we're going to be delivering frequencies depending on, we're going to take a signal, we're going to break it up into frequencies and then deliver the appropriate frequency depending on the sound to a particular location along this cochlea. So how well are we doing? So this is a recent review. And it's interesting. It's looking at a variety of different manufacturers, a variety of different cochlear implants. And what we're seeing here is on the right is the sentence recognition. How well are you doing? So normally hearing humans are the 100%. So interestingly here in the early 90s, we kind of maxed out. We got to about 80% sentence recognition, and there it stayed up until now. Nothing's changed, but another interesting thing is the amount of research here looked at where we're looking at the number of publications that are published every year is sort of dramatically going up in the 2000s, and yet we're not really impacting this plateau much at all. All right, so if this is good, what's bad? So what's bad is tonal languages. Tonal languages are bad. So here what you see is the average correct is not 80%, but closer to like 60. Okay, so tonal languages are not good. music appreciation is horrible. What I've heard from cochlear implant patients is and actually what I've seen from psychophysics is hearing music is sort of akin to taking 3 fingers and basically hitting the piano and trying to appreciate music that way. It's not good. Okay, the other thing that is not good is hearing in a noisy situation. So here's normal hearing subjects, and here's signal to noise ratio. So the smaller this number, the more noise you have, right? And so what you see here, for instance, is for normal hearing subjects and 6 dB of noise signal to noise ratio, we're doing really well, right? You take a cochlear implant subject and you put them in a situation where they're at 6 dB, and they are clearly not doing well. They're getting less than 50% of that sound. Okay, so they got to be in a very quiet situation in order to get this 80% of recognition. Okay, so why is this happening? Why could this be going on? Right. So this is the most mature implant that we have now on the market. Okay. Okay, so here's let's look at the anatomy a little bit more. So here's the cochlea. It's bisected by this basilar membrane, and then in there, sitting right on the basilar membrane, is something called organ of Corti. Now, this basically this particular organ, what it has in there is hair cells, and these hair cells are getting the energy from the vibration of the basilar membrane, and then convert them into neural signals through the synapses to these afferent fibers that go to the brain. Okay, these are the cochlear nerve fibers. Okay. And now let's look at how these fibers actually respond to the neural signal. So let's give it 100 hertz input, right? So the 100 hertz, if you record from one of these neurons, they don't just fire. They fire actually in phase with the changes that you see on this basilar membrane. So at 100, so if you give it 100 hertz, you're actually going to get 100 hertz of response on an individual neuron. Same kind of thing happens at higher frequency. When you get to really high frequencies, each individual neuron cannot, of course, fire at this high spike rate, but the population of neurons still exhibits this very much phase-locked response. So as the brain listens to a combination of these neurons, it still gets this phase-locked, a response that's phase-locked to a particular frequency. Okay, so this is what's called rate coding, right? So there's spatial coding, which is which neuron is firing. Rate coding is how quickly this neuron is firing. Okay, so in addition to this complexity of firing behavior, we have 3 types of neurons that are connected to each hair cell. Okay, these are called low spontaneous. SR stands for spontaneous rate. So these are low spontaneous rate neurons, medium, and high. So these neurons carry some spontaneous activity as they're just sitting there. Okay, now their behavior is different. These the high spot neurons, for example, at low stimulus levels, they carry spontaneous activity here denoted by they're about 50 spikes per second. And then as the amplitude of that signal goes up, they're almost event detectors. So you know, by the time it's normal speech level, it's about 60 dB. What you see is these things are just firing away. They're basically saying, hey, I got something, and that's it. That's the only information you get. Where you get a lot more information is from these low spot neurons, which have a nice kind of a slope. And so if they're firing at higher rate, they're kind of encoding more of the amplitude of that particular sound in that frequency range, wherever they're located. Okay. So another thing I want to point out here is the discrepancy in size. And so I found this really beautiful dissection. So the electrodes are about 100 microns in diameter, and they're huge. So neurons themselves are really tiny, like five microns each. And so you're talking about 1 to 2 orders of magnitude difference in size, right? That's what you've got. So you're not targeting any individual neuron here. You're targeting a lot. They're all together. All right, so that's the problem. On a microanatomic scale, the cells that need to be stimulated carry complex coding. The high spont neurons, medium and low spont fibers are not spatially distinct. They're all together. For us to do this right, the prosthesis really should distinguish between these neurons and be able to stimulate them selectively and evoke very specific spike patterns on these neurons. High spont neurons respond very differently from low spont fibers. So this is very much analogous to what we just saw with a visual prosthesis. Okay.
Dr. Fridman, just making sure I'm understanding this here. So the difference in the high, medium, and low spontaneity rate fibers is what permits for the difference in rate codes that can come from the same spatial area. Just making sure.
No, so the rate coding is still rate coding.
Okay.
That's happening there, right? But depending on the amplitude at the particular region, you might have, they're still phase locked, right? But some of them may be more phase locked than others.
Okay.
Right. You might have these low spont, like over here, if you have a low spont fiber, you might actually get every other spike, right?
Mm-hmm.
But the high spont fiber is just going to be firing away, basically.
Okay.
Okay, so that's cochlear implants. So now let's look at sort of the most new device that's currently in investigation. So this is a vestibular implant. So what does the vestibular system do? So the vestibular system it's an organ in your inner ear. It sits actually very close to the cochlea. And it has these 3 semicircular canals that are filled with fluid, and the fluid serves as sort of an inertial mass. And so when you turn your head, that fluid wants to stay still, and it deviates a little membrane. And what that does is it tells your brain which direction your head is moving in 3 spatial orientations, either spatially distinct orientations, right? What that does, the most obvious thing it does is something called vestibular ocular reflex, and that moves your eyes opposite to that rotation. So if you move your head to the right, your eyes move to the left to keep your eyes on target. So, so this is the biological version of image stabilization that you get on your phone or camera, right? This is the original version. So for people for whom this is broken on both sides of the head, they can't feel the head rotation, and as a result, what they feel is nausea, and the eyes can't focus. So there's a blurring of the visual field. It's not good stuff. So what would be good would be to put a prosthesis in there that senses head rotation. So we have a gyroscope somewhere, senses the head rotation and stimulates those neurons to give them the sensation of head motion that they would otherwise have. Okay, one thing I want to point out that's important here is there are 3 branches to this thing. They're kind of hard to see in this picture, maybe, but you can see one, and each branch of this vestibular nerve goes to a different canal. Okay, And so if we position these electrodes close to each branch, the idea is that we will give the sensation of head movement that corresponds to whatever that particular canal is sensing. So this is what it looks like. There's a processing unit that the patient wears, and then there is a head-worn unit that sends the information to the implant that tells it where to stimulate. Okay, and then the gyroscope is actually sitting in that head-worn unit. Okay, and then this is two different views of the same implant positioned in the labyrinth. Okay, in the vestibular labyrinth. What you're looking here is an ampulla, and the ampulla is the sensing organ. So you have this bisecting membrane, which is a cupula, and as it deviates, there are hair cells down here that feel that deviation and then convert that to neural signals that go back. Okay. So let's look at the performance. So the performance is this is recent. This is something that's going on right now. In fact, there is a patient human trial going on right now. Celia is actively involved in that. So let's kind of look at first of all what is good. What is good is the direction of sensitivity. So you can tell it which direction the head is moving, and it's pretty and it's reasonable. So, and what you can see here in the first row is the blue lines denote, so different colors denote different canal stimulation, or different colors denote the direction of eye motion in the coordinate system of the canals. Okay, so you have the three spatial orientations. And so if you're going to stimulate, let's say, one of the this right anterior canal, you should get a nice eye motion in the opposite direction in that canal. So it should be a big blue motion here, right? And in this case, in B, you're stimulating the horizontal canal, so you should see a big red motion. And that's kind of what you see. And the artifact of sort of the current spreading to the other canals, it can be seen in the eye motion that corresponds to the direction that's appropriate for the other two canals that you're not trying to stimulate. So what you see is kind of a nice, it's a nice direction, right? And here you can see these directions put on a sphere in 3 dimensions, and the expected response are these dashed lines. And so what you can see is the responses are actually pretty good. They're telling you which direction your head is moving. So that's very good. The quality of life metrics, which is something that I did not put here in great detail, but they're clearly beneficial. There was a study that came out in New England Journal of Medicine, clearly indicating this is a good thing to have. Now let's look at what the problem is. So here's modulation depth along the x-axis. Modulation depth, 100% modulation depth is equivalent to 400 degree per second head movement, right? So you move your head really 400 degrees per second is really fast. So you move your head very quickly. Your eyes should move back very quickly at the same speed, right? If it's going to keep up. But the highest speed of eye rotation that we can get with this prosthesis, these are 4 different subjects here, okay? 4 different subjects, and the highest rotation that you can see is something, it's no bigger than, the highest one here is 60 degrees per second when you're trying to elicit 400. Okay, so this is, you move your head at 400 degrees per second, your eyes are basically barely moving to the right, in the opposite direction. So maximum velocity is very small, very difficult to elicit these high velocities. The direction of responses very strongly favors the implanted side. So look, in these in this plot here, this is actually a sinusoidal movement of the head, right? So you're doing this, okay? Now, for one direction, it's very strong motion, but as you're moving back, like here's horizontal, for instance, in red, you're moving this way, in that direction, it's very strong. You move the opposite direction, there's almost no motion, okay? So this is the bad, all right? So why does this happen? So here's a little bit closer description of one semicircular canal, again with a cupula bisection. And as you turn in your head, it deviates this cupula, and you sense this direction. Okay. Now, again, here's the cupula. Here are the hair cells that sense this deviation. Now, let's look a little closer at how this motion is actually encoded in more detail. So what you have here are two different kinds of hair cells. So you have these calyxial cells, which are, they're sort of shaped like flasks. And these are type 1 hair cells. And then you have these other cells, type 2 hair cells, that are sort of cylindrical looking cells. And then you have afferent fibers that are receiving information from these hair cells, and they behave differently depending on which of these hair cells they are synapsing to. There's some that synapse to only to calyces, and then there's some that synapses only with these Bouton connections to the type 2 hair cells, and some synapse to both, okay, dimorphic as they're called. Okay. And so the ones that get information from type 1 hair cells are referred to as irregular cells, and these are called regular. We'll see why in a minute. So here's the response of the regular versus irregular fibers. This is the sinusoidal modulation of the head, and here's how they respond. So this is kind of case in point, because what you have here is, as the head is moving, right, this one thing that happens is the modulation here of the regular fibers is not very big, right? It's kind of moving up and down not very much as far as the firing rate is concerned, and it's very much aligned with the phase of the head motion. Something very different happens with irregular fibers. So they're clearly not phase aligned with a head movement, and their modulation is much bigger. When you're looking at these, at the responses of these two fibers, when nothing gets stimulated, they have spontaneous activity. That spontaneous activity is around 60 spikes per second, which is why it's kind of centered at 60 on both. But the firing is very irregular, meaning that the distance between neighboring action potentials changes quite a bit for irregular responses, and for regular fibers, they are very the distances between individual action potentials is very regular. Okay. So again, so coming back to this, on a microanatomic scale, the cells that need to be stimulated carry complex coding. Regular and irregular fibers have significant spontaneous activity, and regular and irregular fibers are not spatially distinct. They are, again, located anatomically all together. They're sort of wound up. They're not in two different strata. Prosthesis needs to distinguish between these and stimulate them selectively to evoke appropriate spike patterns. I'm hoping I'm getting to my conclusion here soon. Okay, let's look at somatosensory just for kicks. Okay, so somatosensory, you're getting information from a lot of different possible inputs. Okay, there is a huge variety in your skin. And that does not even talk about the proprioception from your muscles, that go back, and of course, there are efferent fibers also going through this nerve. So if you can imagine trying to evoke, let's say, a sensation of temperature by going into this nerve. Anyway, so that's kind of where I'm going. Like it's very, very difficult because these fibers are not distinct, they're all co-located. And that to me represents the biggest problem. Okay. So here's the common success as I see it. Common success is that neuroprostheses are really good at telling the brain which part of the body is getting stimulated. You take this 9-volt battery, stick it on your tongue, you know it's your tongue, right? You put it somewhere else that's sensitive, you'll know where it is. Sometimes that's sufficient. So if you apply this to some location along the cochlea, it's going to be interpreted as a tone to the degree to which it can interpret it as a particular frequency, just based on its location. In the retina, same deal. You put it in a specific location, the brain will interpret that as a blob of light in that location to the degree to which it can. If you put it on the heart and you stimulate, heart is going to interpret that as simply being a signal to it's almost like a binary signal to go ahead and start the electrical network of the heart and do a compression. Okay, so it's like an initiation thing. Again, location is what counts here. Okay, so this is how this whole thing basically got started, right? So the question is what's going on? So before I kind of go into the possible solutions to that, let's also examine a little bit of what individual pulses do, because it's not just the location, it's also what kind of patterns you elicit on these fibers that's important, because what you see here on the right is only a piece of the story. In reality, this is the work that came out of our lab by Cynthia Steinhardt. And so this is work that where we tried to figure out what do pulses do given a pulse rate, pulse amplitude. What kind of firing rate are you going to get? And all of this, by the way, there's experimental evidence for all of this. This is a simulation, but there's experimental evidence for each one of these, and the work that we did looks at the underlying physiology of why this happens. But the bottom line is, let's say you're giving a low amplitude stimulus, right? Low amplitude stimulus reaches what people would at some point call a threshold, but threshold is kind of misleading thing, because what can happen is even at low amplitudes, where threshold means I give a pulse, I get an action potential. But what if you give 2 pulses that are below threshold? Well, if you give 2 pulses that are below threshold, if they're close enough together, they're going to add and then cause an action potential. And so that's kind of what happens at these low amplitudes. You can actually get responses, right? This is what most people think about when they think about stimulation. So past some amplitude, every pulse will start evoking action potentials until, and imagine your pulses are getting closer and closer together. At some point, these pulses are going to overlap where an action potential elicited by one will produce an after hyperpolarization in that cell and not allow a second one to deliver an action potential. And at that point, you have this big drop that's going to drop it in half. And then you have this very complex looking response that comes afterwards at higher pulse rates. At very high amplitudes, you're actually inhibiting this neuron from firing. Okay, now imagine that you have a stimulator and then you have a population of neurons. So the neurons, and now you have high amplitude of stimulation that you're barraging this thing with. At high amplitudes, the neurons that are very close are going to be experiencing this kind of behavior. They're actually going to be suppressed. The ones that are a little further away are going to be doing something like this, and the ones that are really far away are going to be doing this kind of a thing. So it's very difficult to predict what these neurons are going to be doing with pulses. Okay, actually, the whole situation gets a lot more complicated when these neurons start having spontaneous activity, and the whole effect of introducing pulses has actually diminished significantly. Okay. So the fundamental interface problem is this. On the microscale, several types of neurons are co-located. They differ in their firing properties and information coding patterns. And this was, I kind of went through these three different examples to show you that. To encode more than just spatial location, we really need to access specific neuronal types. And we need to talk to them in the code that they're supposed to be delivering to the brain if we're going to get any kind of fidelity beyond place of where they're actually stimulating, beyond this buzzing sensation, in a sense. So here are a few possibilities. I'm getting kind of close to the end. So one possibility is, what happens if you have a whole lot of electrodes, right? People have tried this. Let's make a whole lot of very, very tiny electrodes and then stimulate with very tiny amplitudes. If you do that, you're localizing the effect of stimulation, right? And so now presumably you can get down all the way down to these individual neurons. So the highest resolution that I have seen in the Stanford photovoltaic subretinal prosthesis, which is actually fabulous work. I'm really impressed by this whole thing. Very interesting. But the idea here is you have these stimuli that are these electrodes, but these electrodes are on the order of 50 microns, right? Each individual one is like 14 microns. So we're getting close to an individual cell level. The other thing that works.
Dr. Fridman, I would just like to interrupt that. There is the NeuroPixels probe, which is, I believe, quite a similar resolution to this Stanford probe that we have here.
Yep. Yeah, that's right. Used in the cortex, and I haven't seen any stimulation from that. I know it's used very nicely for recordings.
It does have stimulation capabilities.
Okay. Yeah. Good. Yeah. So that's right. So I'm in agreement with you.
I'm just letting you know if you have options. Okay.
That's true. So for the retinal, this is what I've seen, and so where does it work? So here's the thing: if you take this electrode and you stick it on ganglion cells, I don't think that's going to work very well, because you're not really solving the problem of which neurons are firing. You're still going to have a problem of innervating either an on cell or an off cell at random. And I don't think you've solved the problem. Where it does work is if you go very far upstream and you're talking to in this case, you're talking to the photoreceptors directly. And so you're stimulating just the photoreceptors, and then all of that information gets integrated. That's where it would work. And I think that's where they stuck it. It's a subretinal prosthesis, so it goes right under right next to the receptors. And the other place that it might work is on the cortex. And the reason for that is really the cortex tends to kind of lay things out. And color information could be in one location for a particular location in space, and you might have it sort of integrates these things that used to be all together. It integrates. It actually splits them up into locations along the cortex, and so it might actually work for cortical stimulation. This kind of a tiny, very localized stimulation. Actually, a couple of days ago, we had Lee Miller give a talk, and he basically had a similar kind of an idea for the proprioception stimulation, for introducing proprioceptive stimulation into the brain. Here's another possibility. Some neurons prefer stimulation pulses of specific parameters, so pulse with a pulse rate, so you could sort of dial in on the kind of neuron you want to talk to. And the reason for this is because the different neurons have different channels in them that have different dynamics. And so as a result, you can, in fact, elicit different responses from different neurons. In this particular example, it's using different pulse rates, right, different pulse amplitudes. Another one is everybody's favorite epigenetics, which has become really popular in the past 10, 15 years. Really a phenomenal invention. Should win a Nobel Prize, in my opinion. But the idea here is that if you know what a particular neuron type, what kind of channels it's expressing that's different from another neuron nearby, you can give it these channels that are light sensitive. And so when you shine a particular frequency light on over the entire area, only those neurons are going to respond because they have that particular light sensitive channel. So this was, of course, a great idea for putting into the eye, since the eye is looking at light. It works, but there's additional problems. The problems associated, for instance, with dynamics. So what you see here is here this boy is moving on a skateboard or whatever, and if you have these slowly responding channels, you're not going to see them. So so anyway, it has its own problems, but it's a nice direction to go. Okay. Magnetogenetics. This is another interesting idea. So similar idea, you basically stick this is a particular protein that is specifically sensitive to magnetic stimulation, and then you can dial in on the particular neurons that you want to excite by introducing a magnetic field or a magnetic pulse. And so this is a paper that came out in 2018. Okay. So finally, I'm gonna put something on my work. This is what we do in my lab. So we have this freeform stimulator. This is a device that basically allows you to deliver any stimulus safely. So instead of just having pulses, you can deliver a direct current, or you can deliver slowly modulated sinusoids. And what we found with this device are right now, at least, are two things. One is that we can selectively block pain fibers in a nerve, which is really kind of cool. And so that's it's a selective stimulation. So you're not touching all the rest of these things. In a vestibular nerve, another very cool thing happens is you can maintain the stochastic behavior of these neurons. So if they were irregular, they will keep their irregular behavior. If they were regular, they're going to keep their regular behavior. And what we found is behaviorally, with vestibular ocular reflex, you can get actually much larger eye motion that you would than you would with pulsatile stimulator. So that's what we have there. So to kind of finish it up, I want to basically say that, maybe we should start thinking about how to solve the interface problems independent of application, to kind of come up with potentially new ways of interfacing that we have not yet thought of. Although specific applications, there's a lot to be said for, for specific neurons in that particular area and how we should target them. But maybe there is a more general solution. It could be an inspired method. Maybe we can have electrogenetics where, we introduce some kind of a virus to make particular neurons that we want more sensitive to electrical stimulation than others, if we want to target those, for instance. Lower threshold, or it could be a combination of these various methods that maybe we could use to either sensitize neurons to input and then stimulate them with light or something else. So that's where I am. Oh, Palm was asking you a question I missed. All of your examples of peripheral levels, same pros, still more central. Any pros and cons with more central structures? So my opinion on the more central is, there is complexity clearly that happens in more central structures. I think that because of our ability to do computation and the level at which neuroscience is today, we may be able to guess, okay, what is supposed to be happening in those cortical structures, and it may be actually easier to get the kinds of responses that we want in the cortex, because everything's kind of laid out, as I said, rather than intertwined as things are in the periphery. So it's almost like at the periphery, things are kind of laid out, right? The hair cells are kind of out, and then everything kind of combines into bundles, and then goes to the thalamus, kind of unwinds, and then fans out. But all of this, but of course, encountering all kinds of processing in between. So we have to account for all of that and understand what it is that we're doing at the cortex. But I think it may be, as far as the stimulation is concerned, I think it may be easier in the cortex, but I think that remains to be seen. And that's all I have.
All right. Well, thanks so much, Gene, for the talk and giving us, I guess, a good intro to a bunch of different topics and kind of synthesizing their relations. So we'll open it up for questions. Those of you guys who are still online will see that there's a poll that we just got started. So please fill it out. It'll help us know who attended and kind of where you're coming from. And while the poll is running, if people want to jump in and ask any questions, please do. And yeah, go ahead.
Gene, this is Ed Connor. So you talked about, endless miniaturization of electrodes. But there must be some even theoretical limit to how selectively you could address neurons that are packed close together in cortex or periphery.
Sure. I mean, yeah. I mean, so one thing is, so one thing you don't want to do is you don't want to have big pulses coming out of small electrodes, because that's dangerous. Safety issues, right? But if the whole point is to stimulate one or two neurons nearby, you don't need to. You just need to be close to them. So I think that's the idea, and then you're just modulating that. I mean, I don't know that. I think the main problem that I see is if you're going on that scale, then you're gonna have. It can't be a bed of nails, right? If you have a bed of nails, like I can lie down on a bed of nails and have sleep, right? Why? Because it's not gonna go through me. But if I have one nail, I shouldn't. So the thing is, it's going to be really hard to do that, in that kind of format. And the fact that, some people are looking like, I know Ralph, right? Ralph Etienne Cummings is looking at, positioning very tiny structures in the brain. I don't know how. It's kind of absurd to think that we can make pieces of metal that are going to be stuck next to each individual neuron. There are other ideas out there. The idea that kind of captured my mind at some point is growing a nerve into the brain. So basically it's a little crazy. The idea is you would sort of grow this nerve, and it would sort of connect to individual neurons in the cortex, for example, then would come out, and then you fan it out, and now you can talk to the individual neurons that way. But I think it would need to be some kind of a combination of these things. I don't think it's possible to do it. I mean, I think you can do one, right? But then you have problems.
You know, of course, conduits exist for nerves, and it's been going on for a while, but for the brain also, sorry, I can't remember the name, but, you know, for many years he worked on it with the little conduit or little capillary, let's say, and having growth factor and drawing axons in it, closer to the axon neurons or closer to the electrodes. And in the end, it petered out because you're not also getting whatever perfusion, glial cells, scarring, you know, all those things. So that idea in itself is not as simplistic. And on the scale side, you know, it's worth adding a lot of the nanofiber work, which have even now in the last few weeks come out in major journals, because they not only provide the fine structure, but they can be pliable, so which is good for the brain, because otherwise leads to shearing. And what people overlook, there's a lot of focus on glial cells, but microvascular damage. So there is argument to be made with these kind of nanofiber, like very pliant electrode array. There's a lot going on. So
Yeah.
you know, you did a great overview. This is just a couple of additional pieces.
Yeah, no, I agree with you. Yeah, there's a lot of great stuff kind of coming down the line. I just think, yeah, I think a combination of all of these things needs to be looked into, and I think this needs to be sort of the big effort in neuroengineering going forward. If we're going to be, you know, if we're looking at really high fidelity type of things, and not this leveling off that happened with cochlear implants, where we kind of hit a plateau and we go, okay, well, that's it. So I think that's where all of the effort needs to go. I'm seeing something in the chat. Okay. One issue I see at that scale, even slight motions of the electrode can get in the way of specific targeting. Yes, that's true.
Well, you know, maybe it's, at least for cortex, when we were doing that for the motor, it wasn't as much problem, partly because you're really decoding population. So it's kind of going in and out.
Right.
So in some way, yes. On the other hand, any given day's monkey's optimal decoding doesn't necessarily carry over to the next day. So there's something else going on, right? So there is this micromotion. The vast majority of research has focused on reducing the gliosis, scarring, perhaps some less so vascular, and then least of all is the micromotion. So it's an issue, but you know, all decoding is being done with population, so maybe not as much as you think.
Yeah, no, that's true. And that's, I guess that's the other, so the complexity in the brain is the fact that it's a network of stuff happening. It's not single neurons anymore.
Also, there is a hardly little somatotropy. You mentioned Miller's thing. You know, it's the same thing. There isn't necessarily high neuronal level. So it's not as if like somehow you have a nice, wonderful array will pick off 5 fingers. You know, it's so therefore you're really using whatever we used to use recurrent neural network, but now there are better methods which will have to be tuned to a particular task. And then the neurons themselves seem to retune themselves depending on the task. So I think the complexity is there, and you just, for population recording, giving enough array electrodes helps. And to do enough electrode, the idea preferably is to not do as little a damage as you can. So it's kind of find that nice balance.
Well, and there is also, I mean, there's also the complexity in the cortex because of the fact that you have it's a very dynamic situation that's happening there. None of it is just kind of sitting and going, hey, you know, this is, and there's an actual dynamic loops that are going on wherever processing is happening or not happening. I mean, so you need to account for all of that. And I guess the point I wanted to make is we have to focus on on the neural code, and we have to focus on being able to convey that neural code to specific neurons that are looking for it. And that to me is the big challenge in neural engineering.
If I can emphasize on your behalf, I think you are tackling, everybody's tackling all this population and electrode array and so on. I think you're tackling the harder, one niche and important problem is whether you want to call it DC stimulation, whatever it is, to how do you create inhibition electrically, right? Everybody can excite, but it's not easy to inhibit. And other one, I don't know how far you've gone, but the problem is selectively targeting large and small fibers, because the electrical stimulation as it is happens the inverse. So it does the wrong thing for you.
That's
And
right.
so in that sense, that problem takes new direction. You mentioned pain in passing, although you didn't go into it deeply. So I think that's an example that you want to selectively stimulate large and thin and small fibers. And that's what your work is, and it's great.
Thank you.
All right. Well, if anyone else has questions, feel free to come forward. Otherwise, thank you everyone for joining. This is the first of a hopefully monthly series that we'll be running, and there will be other events on the way that we'll announce. So if you want to hear about those, please join our listserv on the TNT website, which we put in the chat. And if you'd like to kind of learn more, get involved, feel free to reach out to me or Celia. Our emails, again, are in the chat. So we're always happy to get more people involved, and anyone who expresses interest, we can always kind of come up with new ideas and good ways to engage the community. So other than that, thanks everyone. Yeah,
I do have one final question that I like to pose while everyone is still here, and I know this is probably the this is one of those really hot button high up there. So what would you say is, this is kind of addressed, I guess, to everyone that's working on this, what would you say is your working theory for the differences in, recordings from the same neuron from one day to the next, disregarding, the fact that we're even having issues trying to trace a single neuron from one recording to the next?
Well, generally, so there's different schools of thought on that, but I think the dominant one, as far as I can tell, is single neurons are not really representative necessarily of what's going on, and you really kind of have to look at a population if you're going to determine something. And so that's where generally, recordings of local field potentials or multineural recordings are very useful. It's kind of, neurons tend to be sort of statistical in how they function in the cortex. It's kind of a network, and it's a random network.
If I could speak up for stability and meaningfulness of single neuron signals, though, some people may be familiar with the work from David Leopold at NIMH with Bondar electrodes, and in particular inserted into high-level visual cortex, especially especially face-sensitive patches. And the Bondar electrodes have incredible flexibility because they're like 5 nanometer wires that just splay out like the fibers of a paintbrush, and then I think move with cortex rather than rubbing against it. And they easily hold, so they can tell they've got the same neuronal line by fingerprinting with responses to 200 stimuli they show every day, and every neuron has a particular profile of responses across those 200 neurons. And they hold neurons for weeks and months that keep the same fingerprint. So I don't think it's simply the case that single neurons change how they encode information across time, or that they're impossible to address across time.
All right, thank you.
All right, I think that's probably it for today. Again, thanks all for joining, and we hope to see you guys in a future event.
Thank you.
Thank you all. Have a good day.